Good morning to all, dear ladies and gentlemen, esteemed guests, and valued participants. I warmly welcome each one of you for today's webinar. We are delighted to have you join us for this illuminating session on the cancer drug discovery, a molecular journey to targeted treatment. I am Dr. Payal Mittal, co-convener and associate professor at University Institute of Pharma Sciences, Chandigarh University, Mohali, Punjab. We have come together virtually to explore and discuss the webinar entitled as Cancer Drug Discovery, a Molecular Journey to Targeted Treatment. This webinar is designed to be an enlightening and interactive experience, providing you with valuable insights, expert perspectives, and actionable takeaways. It's our 31st webinar being hosted by the Society of Pharmaceutical Sciences and Research, SPSR, in collaboration with University Institute of Pharma Sciences, Chandigarh University, Ghadua, Mohali, Punjab, under the mentorship of Dr. Shashi Alok, National President, SPSR, and Assistant Professor, Institute of Pharmacy, Bundelkhand University, Jhansi, UP, India. Our aim is to ensure healthy lives and promote well being for all at all ages. Our objectives include to hold conferences for promoting the causes of profession of pharmacy, to continue the programs and services that emphasize the health needs of India and the prevention of diseases, to honor eminent pharmacists engaged in the encouragement of pharmacy profession, to promote and facilitate the acquisition, dissemination, and exchange of pharmacy knowledge and information about the practice of pharmacy and pharmaceutical sciences. Throughout this event, we are honored to have an incredible speaker, Professor Dr. Shashank K. Singh, Senior Principal Scientist in Pharmacology Division at CSIR, in Indian Institute of Integrative Medicine, IIM, Canal Road, Jammu. We are also joined by our respected Mrs. Monica Sabharwal, Convener, National Secretary of the Society of Pharmaceutical Sciences and Research, SPSR, and the Managing Editor of International Journal of Pharmaceutical Sciences and Research, IJPSR and IJP. Without taking much time, I request Mrs. Monica Sabharwal ma'am to kindly take the lead to introduce and welcome our guest speaker, Professor Dr. Shashank K. Singh. Over to you, ma'am.
Thank you so much, Dr. Payal Mittal. A very good morning to all. I am delighted to welcome you all to the 31st SPSR webinar on the cancer drug delivery, a molecule targeted journey to the targeted treatment, organized by the Society of Pharmaceutical Sciences and Research (SPSR) in collaboration with the UIPS, Chandigarh University, Mohali, Punjab, India. This webinar promises to be enlightening and insightful exploration into the world of cancer drug delivery. I am Monica Sabharwal, the convener of this webinar, and it is truly an honor to understand before you today as we embark on the journey that holds immense significance in the realm of the pharmaceutical sciences. Today's event is a testament to our commitment to advancing knowledge, fostering collaboration, and pushing the boundaries what is possible in the field of cancer research. Our distinguished speaker for this webinar is none other than Dr. Shashank K. Singh, a senior principal scientist and the professor in the Pharmacology Division at CSIR, Jammu, India. The Triple IM Jammu stands as this esteemed laboratories in the CSI Government of India. It is actively involved in the pioneering novel drug discovery programs, drawing from rich inventory of natural, semi-synthetic, and synthetic sources. Dr. Singh's expertise and the contribution to the field have been invaluable, and we are privileged to have him share his insight on this topic. The theme of today's webinar, the cancer drug discovery, a molecular journey to the targeted treatment, is a forefront of the scientific innovation. With the ever evolving landscape of cancer research, understanding the molecular difficulties and developing the targeted treatments is crucial for improving the patient's outcome and advancing in the field as a whole. I am pleased to announce you that we have received more than 3,500 registrations from more than 25 countries. This global participation reflects the shared commitment of scientific community to collaborate and exchange ideas for the greater good. Today, we are honored to have with us today the Dr. Shashi Alok, the National President of SPSR. His leadership and dedication to the scientific research have been pivotal and propelling progress and fostering innovation within our community. I would also like to express my sincere appreciation for the invaluable support and contribution of the Dr. Payal Mittal and the Dr. Sanjay Nagdev. Their efforts have been played crucial role in transforming the vision of this event into a reality. Behind the scenes, there is a dedicated team whose hard work and commitment have been driving force behind the seamless organization of this webinar. Their attention to the detail and the passion for excellence have made this event possible. And I extend my heartfelt gratitude to each and everyone SPSR team member. Finally, to the all participants, thank you for being an integral part of this gathering. Your presence enriches the collaborative spirit of this webinar, and we look forward to the vibrant discussions and insights that will unfold throughout the day. So, without further ado, let's dive into the enriching session. I encourage you to all actively participate, ask questions, and engage in the discussion that will unfold. Together, we can make a significant strides in the fight against the cancer. Once again, thank you for joining us, and I hope you will find today's webinar both informative and inspiring. Thank you. Let's begin the 31st SPSR webinar. Over to you, Dr. Payal Mittal.
Thank you, to allow me conduct this webinar. Let's start with a brief introduction of Professor K. Working scientist in Pharmacology Division at CSIR, Institute of Integrative Discovery from the last 10 years, CSIR, Government of India. Just in the formulation development department and in the in from 2003 to 2008. He has earned his master's degree in pharmacy (M.Pharm) in the year 2000 from Birla Institute of Technology and Sciences, Pilani, Rajasthan, India. He did his bachelor's degree in pharmacy in the year 1998 from Dr. H.S. Kaur Central University, Sagar. He has more than 100 international publications in peer-reviewed journals and 6 US EU patents to his credit. Year 2009 and 10 by DST, Indo-US Science and Technology Forum, New Delhi. His areas of interest are cancer drug discovery from natural resources, products, drug combination studies, targeted drug delivery systems for cancer, and cancer immunotherapeutics. We have received more than 3,500 registrations till now. Registrations are received from India, Indonesia, UAE, Kuwait, Pakistan, Bangladesh, KSA, Nigeria, Ethiopia, Canada, North Macedonia, South Korea, Ukraine, Iraq, Algeria, Myanmar, Poland, Albania, Switzerland, South Arabia, and Nepal. We are overwhelmed to express gratitude for their participation. For joining us today. Highlights of today's webinar include
Dr. Shashank, may I request to please share your presentation? I think Dr. Payal internet may be unstable.
Yeah, there are some issue with the audio. I think let me share you my slides. Are my slides are visible to you?
so
Yes, sir.
without
So shall we start with the presentation?
Yes, sir. Sure, sir.
Yeah, at the outset, I would like to thank the Society of Pharmaceutical Sciences and Research for inviting me for a lecture on cancer drug discovery, a molecular journey to targeted treatment. I would like to thank Dr. Shashi Alok from Bundelkhand University, the organizer, one of the main organizer of this event, Dr. Mrs. Monica Sabarwal, Dr. Payal Mittal, and all other team members of Society of Pharmaceutical Sciences and Research. So, as you know, that the cancer drug discovery has a my slides are visible to you.
Yes, sir. Visible.
But they are not moving actually. Yeah. So if you see the last 1 century, from the beginning of 20th century, the advancement in the drug screening programs and the uh, drug discovery projects. So if you see at the beginning of 1900 century, the Paul Ehrlich, the German chemist who first time who coined this term chemotherapy, first time he was testing these chemotherapeutics against syphilis, the arsenic compounds and arsenals, so in the animal models. So he got some success with the uh, these chemotherapeutic agents. Later he wanted to discover the new molecules for cancer also. He tested some dyes like aniline dye and other dyes, but he could not get much success to that, but he developed some initial rodent and rabbit models for testing these type of chemotherapeutic agent. So if you see the advancement of the cancer drug discovery program started at the beginning of the 20th century. Later on, the scientists could able to develop new transplantable tumor models during 1900 century to 1915. The early Shasite's carcinoma model, you know, that is at the beginning stage of early Shasite's carcinoma, and other tumor models like sarcoma 180 models. They were the preliminary models which were used initially for testing the chemotherapeutic agent. Later on, when that time during the World War time, they used to get the nitrogen mustard as a war drug. So during that time on the naval ships, they were carrying some nitrogen mustard. What they found is that there was a depletion of WBCs. So in the persons or the soldiers, so they got some hint that that could be because of this nitrogen mustard compounds, who basically induce the bone marrow suppression in the naval soldiers. So later on, the professors, pharmacology professors from Yale University, uh, Professor Goodman and Gilman, they first time tested these nitrogen mustard in the animal models. The less volatile compound of the nitrogen mustine, they tested in the animal model, and they found that there was a bone marrow suppression in the animal, and there was a decrease in the level of WBCs. So from there they got some hint, they developed some lymphoma models in the animals, and there they got success that there was a regression in the lymphoma, lymphatic cancers. So thereafter, these type of animal models were quite useful for testing the new novel agents. Later in the America, they came up with the Cancer Act, where they initially set up some new labs, which under NCI, which was NIH, one of the ambitious program, where they established some of the primary screening tumor cell lines, where they tested some of the chemotherapeutic agents initially with in the leukemia model like L1210 and other molecules like initially they tested like some compounds like antifoliates or DFHR inhibitors, like because you know that if you eat the green vegetable, they contain high amount of folic acid, and they basically nourish the tumor cell, highly metabolic cells. They take the folic acid from these nutritional components, and then they proliferate very fast. So they got the hint that if we stop this if we have to stop this proliferation, if you stop giving cancer cells to the folic acid, then there is chance that you can stop the further proliferation of the cells. So there they got the antifoliate drugs like methotrexate and other molecules. And later on, they also discovered the 5-fluorouracil like compounds, which basically are very useful for solid tumors. So that was the initial journey of the cancer drug discovery, right from the 1900 century till 1960. Later, they discovered some new actinomycin D like antibiotics, which are commonly used for cancers. Then Eli Lilly company, they discovered vinca alkaloids, which are basically very useful for leukemia, vinblastine and vincristine, which are very popular compounds. You know that they are commonly used for the pediatric leukemias. So these type of drugs basically came up because of these NCI 60 research program, where they established 60 human cancer cell lines panel, and they start testing these natural products against these type of tumor cell lines. And from there they got some hint, or sometimes they got some new molecules So if you see the history of cancer drug discovery, more than 60% of cancer drug has come up from the NCI, National Cancer Institute of America, and most of the are either have been taken lead from the natural product, or they have come directly from the natural product. So initially they thought that the, as you know, that the chemo cancer treatment is always is you need to have a three prong approaches, like radiotherapy, chemotherapy, and your surgery. So usually initially in those days in 1960 they used to believe that only radicals treatment like surgery and radiotherapy can eliminate the cancer from the body. But due to micrometastasis of the cells in the tissues, they could not able to cure those type of cancer. So later on the adjuvant chemotherapy came into the picture, and thereafter the new treatment models, like we could able to develop some xenograft model, orthotropic models. So these type of new advancement came into the picture, and where you can directly implant the human tissues on the animal, because these new mice, they don't contain bees T cell immunity, so you can directly place the human tissue on their skin, or you can allograft the skin on the skin, or you can directly, or you develop the orthotropic model on these animals, and you can develop the, it can mimic the similar kind of tumor model which is present in the human. So thereby you can reduce the cost of the testing in the clinical trial. You can test the molecule in your laboratory setup, and you will come up with the activity. If you find the molecule has got good activity against these type of tumor models, you can develop the new chemotherapeutic agents. So later in the 1990s the new molecule came into the picture, like more targeted approach of chemotherapy came, like imatinib mesylate or Gleevec, you know, that very popular drug which came into the picture in 1995 to 98. So that was basically tyrosine kinase inhibitor. So that was that was the first drug which was a target based kinase inhibitor. So after that, once the scientists able to complete the whole genome sequencing of through the human genome project, the whole genome atlas is available now. There now you can design the molecules which are more specific to the mutation present in, or different type of biomarkers which are present on the cancer. The new therapeutics can be designed on that basis. So today's my talk will be focused on the more, right from the cytotoxic agents to the target. We shifted from the cytotoxic agents to the target based molecules. The later now we are having more uh, personalized or precise medical care, where you can have precisely target the cancer. So as you know that drug discovery programs comprise of various facets of the this thing, like right from the lead profiling. First you try to identify the hit you try to identify, from hit you optimize the hit to the lead. Then lead candidate can be further tested by doing the lead optimization. You can do all sort of preclinical profiling of the drug. Then further, once you test the molecule found to be active in the preclinical models, you go for the clinical trials. So right from the identification, target identification, as I have told you in the previous slide, that you know the mutation present on a cancer, so that will be your target for a cancer. Because now the treatment is shifting, there is a paradigm shift in the cancer treatment, and it is towards more personalized treatment. So if somebody has been diagnosed with cancer and he has some specific mutation present on his cancer, so that will work as a target. So once you identify the target by doing the whole genome sequencing, or exonic part of the DNA can be sequenced, which is basically a coding part, which basically code for your proteins. So that exonic part can be sequenced, and where you can find out the which type of mutation is present, and because of that which protein has been mutated protein has been expressed. So on that basis, the new targets or new biological assays can be designed, or new antigens can be designed, through which you can these antigen will generate the new antibodies. So on that basis, new assays can be designed, and you can test the molecule, and on that basis, the new more personalized care can be given to the patients. So if now I am if I talk about the drug discovery program at our institute, so there how we go about the drug discovery program is like, we do the bioprospection of the plants, we create and manage the resources, like we have a good amount of natural compound repository in our institute, whether it is from the microbes or fungus sources or plant, or all sort of these things. So we have kept a repository where every individual or every student who is working on natural product chemistry, he has to once he has isolated some pure molecule from his research, he has to submit that molecule in the drug repository library, where is it will be remain in as a active molecule in our drug repository. So later, if somebody want to cross check the activity, he can always go back to the repository and he can test that molecule further. So then we have a validated traditional knowledge digital library. As you know, that CSIR is handling this project, which is basically the validation of TKDL. It is called TKDL, traditional knowledge digital library, where we have all the plants have been catalogued, and we have all the passport data available of these plant material, with all the pharmacognostic information is available on the website. So you can always visit the TKDL website, where you will find all the information related to your research plant material. Then we also do the biology of the secondary metabolite, because you know that secondary metabolites are basically the defense mechanism of plant. So they are produced to protect, but they are also very very useful for human as a if you talk about as a medicine. So isolating these secondary metabolite and increasing the level of um, these secondary metabolite can be one of the strategy to produce the natural product. So you can do the metabolic engineering by manipulating their biosynthetic pathways, by by checking their key rate limiting steps in the biosynthesis of these metabolites, and you can have a better plant strain where you have more amount of your desired natural product. So that type of work also we do. Then we discover the new pharmacophores also by doing all focused library approaches. Then we do the all sort of structural activity relationship, like quantitative structural relation QSAR models we have. So likewise, we do QC, QA, and CMC of the plant material. So that is all about the overall gamut of the activity which we perform. So for any good bioresearch, you need to have a very good you need to grow them in a controlled conditions. So we have a farmland in Kashmir as well as in Jammu, where we have 150 acres of land in Jammu, and around 50 acres in Jammu, and 150 acres in Kashmir and Bonera, where we grow the high value medicinal plants and aromatic plants. So you can see over there we have lavender cultivation is going on in Kashmir. So that is used for many in the fragrance industry, in perfume industry. So that is one of the aromatic plant which is grown over there. Similarly, in Chatta, we are growing the other medicinal plants like cannabis and other mentha and other aromatic plants. So this is the field trial experimental farm where we grow our plant material for a bioresource. Then we have a identify for identification of crude drugs. we have a Janki Amal herbarium drug repository, where we have more than 4,200 of crude plant drugs and plant parts. We have a herbarium specimen more than 22,000, and already CSIR triple IM as well as CSIR as a whole is digitalized the Ayurvedic plant in a TKDL software. We have a crude repository of drug. We have all passport data of herbal drugs, photographs of all drug live plants, pharmacognostic data, and all chemical fingerprinting of these plant material. So if you talk about the conventional process of isolating these natural product, basically it involves lot of steps. If you see, you start, you take the methanolic extract, then you change the polarity of the solvent, like from hexane to chloroform to ethyl acetate to methanol. So by non-polar to polar, you just take all sorts of uh, solvent system, and you try to extract the desired molecules from the planting your crude extract. So these fractions are further tested in the different type of biological activity, and if you find some activity in these fractions, you further subfractionate these fractions, and then you try to isolate the pure compounds from these fractions. But it takes hell lot of time and energy and efforts. So we have a system like SAP box is basically a hybrid system where you have a combination of HPLC and solid phase extraction process, where you can produce more than 576 fractions in a 24 hours of time. So it takes it it basically its complete separation can be done in a day, and it is basically two-dimensional separation, and recovery rate is very high for both non-polar and polar substances, more than about 90%. So by doing this automation, you can have a highly reproducible process of extraction the natural products from the plant material, and you can save a lot of time and solvent by doing this box fractionation. So if we have a targeted drug discovery programs also for natural products, where we have already in our lab we have set up some targets which have got good success stories in the past, like PI3K kinases, EGFR kinases. So you know that there are many kinase family drugs are available in cancer, especially for target based therapy. So we have set up many kinases in our laboratory, where we can test our molecules for target driven drug discovery. So once we isolate the pure natural compound, we try to optimize these natural product by doing the semi-synthetic modifications, and by doing QSAR and medicinal chemistry around them, taking the advantage of in silico studies. We try to modify the compounds by using the Lipinski rule and other rules, so that make the drug more druggable for the targets. And we do all sorts of PK PD tox activity, and in vivo we have good amount of models. We have all CYP assays, like CYP liabilities are tested, like CYP, all CYPs are there. So we test the, we also have the toxicity models, like for acute toxicity, subacute toxicity models. So all sort of preclinical drug development we do for target based drug discovery programs also. So this is the overall activity which I have told you, that we use plant, marine, and microbes, all sorts of natural products. We try to get the novel compounds from these sources, and we test them for varied diverse panel of activities, like antifungal, antimicrobial, anti-cancer, anti-diabetic. So there are many activities which are going on in our laboratory. So we test them, and if we find some hits in that, we try to optimize the hit into lead, and then we do all or generate the NCE by using different uh, chemistry protocol, like mutational synthesis, semi synthesis, diverse oriented synthesis, precursor directed synthesis, to get the optimized lead NNCE. We also do that for conservation and production of bioactive plant using in vitro techniques. You know that there are many plant species which are endangered. So to protect these endangered species, the best way is to protect these endangered species is tissue culture technique. So you can culture these endangered species and produce the new plant. You can do the field after hardening these tissue culture plant in the high humid environment. You can just put it in the field transfer. So that job also we do. These are the target species which are basically, some of them are very endangered, so we try to protect these endangered species by using tissue culture techniques. And we also use the bioreactors where you can produce the secondary metabolite in a larger quantities by manipulating the different culture conditions, and by using biotic, abiotic stress, and other tissue culture, like using different phytohormones and other parameters like oxygen, aeration, carbon dioxide levels. So by changing these, you can manipulate with the media composition, you can improve the production of secondary metabolites. Am I audible to you?
Yes, sir. Audible.
Okay. So as you know that if you see the standard care of treatment is basically like involvement of five type of standard treatment options are there. For local cancer, like if cancer has not been metastatized to the other areas of cancer parts of body, they usually go for surgery and radiation. If it is a metastatic disease, then the surgeon, before going for surgery, he will try to use the adjuvant chemotherapy, where he will give some chemotherapy so that the cancer will shrink and it will come to the original site from where it has started spreading to the other parts of body. So in that scenario, there is always adjuvant chemotherapy is very helpful to shrink the size of tumor and make the tumor more resectable for a surgeon. So that all depends on their call of the surgeon, radiotherapist, and other team members of the intervention surgical oncologist or other team members. Like they have a tumor board, so they take a collective call what should be the first, like whether they should go for surgery first or radiation first, or they should start with the chemotherapy. So for local treatment you have surgery, radiation. For systemic you have a hormonal therapy, chemotherapy, and targeted therapy. So surgery, you know that cancer can be removed by surgically resecting the tumor. And after that, if some, after the RO margin means there is a zero, you will try to remove some non-cancerous part also, which is neighboring to the cancer, because there is high chance that some of the cells may metastatize to the neighboring surrounding tissue of the cancer. So if he will, the surgeon will send the samples to the lab for testing for this thing, the cells. So if he find that, he finds the RO margin, like there is zero metastasis in the neighboring tissues, there is, then the patient is safe. If he doesn't find RO margin, that means that he need to give the radiotherapy also, because there is likely to be spread in the other parts of the neighboring tissues. So then the patient has to receive radiotherapy, and then chemotherapy will be given after radiotherapy. So it all depends on the stage of the cancer. If it is a local cancer, means it is in one part it is present, then it is easy to be resected from the body, and other modalities can be used like chemotherapy or radiotherapy. But if it is a locally advanced tumor, like, and now the tumor has spread to the neighboring tissues also, then there is chance that survival and other modalities will also get involved. So cure rate will be decreased in that scenario. Initial stage, you know that once you there is always a saying that if you diagnose the cancer at the earliest, if you can pick up the cancer at the early stage, there is high chance that you can cure the cancer. But if there is a delay in the diagnosis of cancer, then there is high chance that it may not be curable in that scenario, because it has already spread to the other part of body, so it becomes difficult to treat that cancer. So now, depending on stage, type of cancer, then personal age, health cycle, how much patient, what is the age, like if he is very old person, he may not able to tolerate the chemotherapy or other treatment modalities. If he is a young person, he will be able to handle the harsh treatment, or how many cycles has to be given. So it all depends, the treatment plan is based on all these factors. But these treatments are still not very tailor specific, or cancer treatment is not like usually what still we are doing, that whatever protocols have been developed, they are on the western peoples, or the protocols of the drugs which they have done some clinical trial on other population, not on Indian population. So there is a molecular changes in every type of cell. It depends on the races, it depends on the traits. So it all depends. So one size will not fit for all, because the molecular signature of cancer will be different for Caucasian, for Asian population. So now there is need to have a more personalized care. So for this, you need, now you have a, in addition to chemotherapy, radiotherapy, surgery, now the treatment, there is a paradigm shift in the cancer treatment. Now you have a biomarker testing first. They will test your cancer for different biomarkers. If those biomarkers are present, there will be a precision medicine for you, where you can precisely target the cancer based on the biomarker present on the cancer cells. Because you know that these biomarkers has been identified, because now you have a whole genome atlas is available now. So they can just send the sample of your patient sample for a lab for genome sequencing, exome genome sequencing, and they will find out which are the biomarkers. Like if PI3K pathways, PI3K is mutated, they will give cocktail of drugs of PI3K. So based on that, the treatment modalities can be planned. So similarly, some patient may require stem cell transplant, hormonal therapy, targeted therapy, immunotherapy. So I will not go in all in the detail, but I will try to cover some of the things in my lecture. So you know that biomarkers can play a very vital role, because they are measurable indicators. They tell something about a person overall health and disease state. There can be a theranostic biomarker, because they can be used for a therapeutic purpose as well as for diagnostic purpose. So these biomarkers are necessary to administer effective therapy to the patient. In the old days, also in the 6th century, if you see Shushut and Charak, you can see in the right side of your slide. If you see, there is a ants are moving from this pot. So basically, the presence of sugar in the urine is basically a biomarker for identification of the diabetes. So during that time also, that was the biomarker, which was a crude biomarker, but still it is very important biomarker to diagnose the disease. Similarly, so disease pathological stage, biological processes, response to the treatment, these all things can be very well measured through these biomarkers. Because earlier in Ayurveda also, you know that Vata, Pitta, and Kapha, three major principle are commonly being followed or by Ayurvedic physicians. So they all are governed through the heart rate, blood pressure, and based on these conditions only the disease can be identified. So biological markers or biomarkers are very important for designing the treatment plan. So the new theranostic biomarkers can be very useful for making a personalized care for a cancer treatment. So screening of biomarker can be done through in vitro by fluids, ex vivo through biopsy samples, and in vivo through bioimaging. So diagnosis, they are very helpful for diagnosing the disease, like whether the at what is the risk of patient profile. So selection of the treatment plan response can be measured by these biomarkers, or what will be the chances of the reoccurrence of the disease can be evaluated by using these biomarkers. So image guided intervention procedures, also these biomarkers are very helpful for imaging of the tumor and for radio diagnosis of the disease. These biomarkers are helpful. So the questions can be asked by these biomarkers can be very predictive in nature. They can be diagnostic in nature. They can be prognostic. They can tell you about the pharmacodynamics of the disease and reoccurrence. So predictive means whether the person will likely to develop the cancer in future. So they will tell you, now you know that Genome Patri, the new startup have come up, or there are many others companies have come up. They can they can send your sample to the lab, and they can just do the some common pathways which are basically aberrant in the cancer. So based on that information, they can tell you that that person will likely to develop the cancer in future or not. So that kind of predictions can be made by these biomarkers. The diagnosis can be very easy by using these biomarkers. The prognostic value can be obtained by these, whether the which drug will be the optimum for better response. The pharmacokinetics or dynamics can be measured by this, that what will be the optimum dose for this body and what will be the chances of cancer return. So there are some common this thing like EGFR, KRAS, ALK, BRCA. These are the common therapeutic as well as diagnostic biomarkers which are predicting for treatment efficacy. So radiotheranostic biomarkers, have I already told you that they can be useful for imaging purposes also. So they can be identified. You can identify the stage, because you know that when somebody visit to the doctor, he will say that if he has some doubt that the person has cancer, he will ask for go for PET scanning. So PET scanning, where you have a fluorodeoxyglucose labeled, radio labeled sugar basically. So basically it once they inject this radio labeled sugar in your somebody's body, fluorodeoxyglucose, it emits positron, and basically it will go in the cancer tissues also in the body, and it will flourish there. So that fluorescence will be measured by the machine, because cancer cell has a high metabolic activity inside their cells. So they basically start emitting the high amount of fluorescence. So if you have a more SUV values, because if you see the PET scan report, they always mention the SUV values. So SUV is nothing but it is a standard uptake value. So the dye which has been taken up by the cells, that will be measured. If it is more than 2, the uptake value of the your fluorodeoxyglucose, that means that there is high chance that there is a cancer present in that area. Because of high metabolic rate of these cells, they will have more amount of glucose uptake of these cells, and they will emit more positron. So that can be quantified by the machine. So they can be very useful for staging of the disease, because if the cells are spread throughout the body, so the doctor or radio diagnosis person can easily tell that it has spread to the whole body. So staging, based on that staging, can be planned. What type of cancer can be predicted, what should be the dosing. So all these things are very important for designing the treatment plan. So now you have a better machines like positron beam therapies in the radiotherapy, where you can more precisely control the treatment, because they don't have exit dose. The proton doesn't pass through the cell. So in the radiotherapy, earlier we used to give gamma rays or other X-rays. So they basically penetrate if the some cancer is present, like some cancer which is like somebody is having cancer of say uh, liver. So if there are some vital organ present around liver also, so if some radiation has to be given, the neighboring vital organs will also receive that radiation, and there will be damage to the neighboring tissues also. Or if some healthy organ is beneath the that organ, that will also receive some radiation. So in a way you are damaging the vital organ also with by using the radiations. So now you have a proton beam therapy, which is basically doesn't have exit dose. So it will not pass through the body. So it will just travel. The radiotherapist will make a 3D contour of the tumor, and based on that, he will software will just give the radiation only to the cancer tissues. So in a way, you can safeguard the vital organs also. So I'm just giving you some of the example of some of the interesting experiment we did with the radiation with the chemo in the combination with the chemotherapy, where we gave the IR radiation to the cells, and we produce the DNA damage to the cells, and then we check the P53 level. You know that P53 is the tumor suppressor gene. Basically, it is a DNA repair. It helps in the DNA repair mechanism. So whenever there is an insult to the DNA, there are some repair enzymes and repair genes are there, which like P53, P10. So these type of there is increase in the expression of these tumor suppressor genes. So P53 level will go up, P10 level will go up in that scenario. So which will basically inhibit the PI3K pathway, which AKT-mTOR downstream you have a signaling PI3K AKT-mTOR. So basically we gave two grays of the dose, 3 grays of the dose, and 5 grays of the graded doses to the cells, and we can find out that in the U87 cell and T98 glioblastoma cell lines. Similarly, we gave the same radiation dose to the U87 T98 cell lines, but along with thymoquinone, which is a natural product. So along with that, we combined the radiotherapy with chemotherapy with natural product. So TQ plus 5 grays of the IR dose of this thing. So what we can see is that the TQ or thymoquinone is basically radiosensitizer. So basically it will sensitize the cells so that you have more pronounced cell death when you give prior treatment of chemotherapy before giving the radiotherapy. So in a way, it is basically if you have some drug which basically radiosensitize the tumor cells, so it will have which can be amplified. Your effect of cell death rate will be amplified by using these chemotherapeutic approaches. So by using there are now I will just explain you the precision medicines, like how the biomarker identifications can be done and how precisely you can target the cancer. So if somebody is having cancer, he can just send the sample to the lab. They will do all sorts of target screening, like they will do the whole exome sequencing genome. They will identify the uh, they will do the sequencing of the genome of the tumor, and they will do the transcriptomic data also. They will be generated by using RNA samples, and protein samples can also be used for non-tumor for controlled sample if required, because ultimately what happens is GNA get converted into mRNA, and mRNA finally produce the proteins. So these final outcome will be the protein. So if you do the IHC like immunohistochemistry and other proteomic things, you will come to know that whether there is a mutated protein has been expressed. So in a way, you can cross check the overall pathway. So tumor data will be compared with the control normalized tissues with the normal control and the laboratory standards, like the in the NCBI website, you know that all the data bank is available. So if you know that if you want to check the BRCA1 or EGFR kinase or KRAS, so you will come to know that which gene have been mutated. Based on that, they will identify the biomarker, whether a single nucleotide variation with respect to control sequence, or whether there is in indel, or where there is a deletion of the sequence, where there is a there where whether there is a micro satellite instability is there. So that type of data can be obtained. There is, if there is an epigenetic modifications has been, like whether there is a hypermethylation to the promoter sequences of the gene is there. So these type of biomarkers are very helpful for designing the newer treatment protocols. Similarly, like PDL1, you know that the last 2017, they discovered that this is the immunotherapeutic marker PDL1. So basically, a programmed cell death ligand receptor 1. If it is present in the cells, then the patient can qualify for the immunotherapy, otherwise not. So this is the NGS pipeline where you can just the sequencing can be done, DNA can be fragmented into library, and then it can be enriched, and then gene probes like KRAS and other mutation can be reached. You can also check with the limited PCR by enriching these libraries, and these paired and next gen sequencing data. You can just check with the exonic part of the DNA, where if the you are using for KRAS mutation, so you can do the alignment with the reference genome, and you can identify the binary alignment files. So these clean BAM files will further tell you whether there is a single nucleotide variation is there, whether is incision or deletion is there, or there is translocation of the whether there is a translocation of the translocation is there or not. So these are the common panels which are commonly used for the detection of the mutation in the cancer. So I will just talk about today about the KRAS mutation, because KRAS is the one of the common cause of the cell death in the colorectal cancer. So these are the common panels which are normally used for the precision cancer care, like ALK, BRCA1, BRAF, EGFR, KRAS. These all are the panel of the mutations which are commonly present in the disease. So predicting the biomarker for immunotherapy, as I have already told you, that for the triple negative breast cancer patient, usually if the PDL1 positive is there, they will give pramzolimab, which is a immunotherapeutic drug plus chemotherapy. If PDL1 is negative and there is a visceral crisis, like there is overburdened growth of the cancer tumor cells, so there will be call, in case visceral crisis is there, they will either go for, if it is no visceral crisis, they will go for monotherapy like anthracycline, derivatives can be given, taxane will be preferred. If there is a visceral crisis is present in the patient, then combination chemotherapies or anthracycline derivatives like can be given, or taxane can be given. If the BRCA1 germline mutations are present, then, and if it is the patient is PDL1 positive, then pramzolimab plus chemotherapy can be given. If patient has BRCA1 mutation, PDL1 negative, then olaparib or PARP inhibitor or any other chemotherapy, platinum drugs can be cisplatin or other taxane can be preferred. So these are the predictive biomarker PDL1 receptors or BRCA1. So KRAS is also a predictive biomarker in colorectal cancer, because approximately 40 to 45% of colorectal patients have KRAS mutation. So these are the driver mutations. Basically, they drive the cancer. Or KRAS mutation is not a hereditary mutation, not a germline mutation. So why it is important is because aberration is in the, because of our lifestyle now. You know that we are habit of eating a lot of junk food like pizza, burger, momos, and other type, all sort of junk food. So ultimately, they produce lot of aberration to the genome, and that genome changes in the genome. Ultimately, if some mutation will take place in the KRAS, ultimately it lead to the cancer in the GIT. So the most common mutation in KRAS is glycine codon is at 12th position is replaced with the valine. Glycine is replaced at 12th codon with the aspartate. Glycine at 13th position replaced with aspartate. Glycine at 12th position replaced with cysteine. So glycine. So that whole lot of game will play because cysteine is a non-polar amino acid. So in that situation, the things will change. So accordingly, these mutations are very critical for therapy. So when and how KRAS biomarkers are tested, so at stage 4 or metastatic colorectal cancer, these are tested, and RAS status must be determined prior to start of anti-EGFR, because erlotinib, which is a EGFR kinase inhibitor, but you cannot give directly to the colorectal cancer, metastatic colorectal patient, because we need to test them whether these KRAS mutation is there or not, because you know that ultimately the if you see the EGFR kinase pathway, the downstream signaling pathway where is KRAS is present. So if you give EGFR kinase inhibitor like erlotinib, so ultimately it will not lead to the final stage, because in the middle there is KRAS is sitting. So the pathway and the downstream you cannot go further. So because of the mutation present in the KRAS, so you will have a very poor response in EGFR therapy because of the mutation in the KRAS. So they are very predictive in the nature, because colorectal cancer cannot be treated with this EGFR kinase if mutations are present in the KRAS. So in the KRAS, 40%, if you see the overall status, 40% of mutations, 40% of colorectal cancer have KRAS mutation, out of which 63.5% have G12 mutations and G13. So similarly, this is, if you see the pie chart, you will come to know that which part of codon is basically majorly playing the role in the colorectal cancer. So basically what happens is KRAS, this dynamic switch, when GTP bind to the KRAS, it activates and it get hydrolyzed. So fast hydrolysis of the GTP can lead to activation of the KRAS. So these exchange factors are basically very critical in the cells for GTP binding to the KRAS and hydrolysis of the KRAS. So if you see in the screen of your left side, there is a wild type of KRAS, and in the left side wild type, and the right side you have a mutated KRAS. So if you see the RAS signaling, if you see the upstream signal, there is a EGFR kinase, and it goes down. If you see the downstream, there is KRAS, and there is a MAP kinase pathway, and there is PI3K kinase pathway. So ultimately, both give different type of signal. One is survival signal, one is giving proliferation to the cells. So if GTP is binded to the cells, the signal will not pass through, and then ultimately it will lead to downstream signal will not pass through it. So transit activation of the this thing will, in the normal cell, it will only transiently it will be activated, but in the cancer cell it is chronically GTP bounded and activated and dysregulate the signaling. So it will, GTP will always be binded to the this thing, and it will keep activating the cancer cells to proliferate through PI3K signaling downstream pathway. So if you see, there are different type of inhibitors like alpelisib, mTOR inhibitor, tacrolimus, everolimus, and there are many other drugs which are under clinical trials. There are many natural products which are Kras attenuators like curcumin, epigallocatechin gallate, epigenin, quercetin, lycopene, ursolic acid. So these are capsaicin. So these all are natural products. They basically affect the RAS effector activity. There are some molecules like quercetin. They basically works through the RAS processing. Some are through the RAS membrane association. So there are many natural products which take part in the attenuation of KRAS. Lupiol is also there. So there are many natural products also which are basically have a different kind of mechanism through which they regulate the KRAS attenuation. In the beginning, it was believed that the KRAS is not a druggable target now, but you have a sotorasib, the drug which is basically target the KRAS. So today I will be talking about one of the our molecule which we tested, which is basically a bosphilic acid derivative, which is targeting the KRAS G13D, which is a predictive biomarker. So you know that it is contain bosphilic acid contains major triterpenoids like olein and orsen, and their molecular target are BAXBCL2, another AKT PI3K signaling pathway, and there are many clinical trials which are underway for testing these molecules. So we synthesized some semi-synthetic library of 31 compounds of bosphilic acid, beta bosphilic acid, where we found that our one of the molecule has showed good activity in colorectal HCT116 cell line, which is G13D, which is harboring the mutation at G13D, colorectal cancer in the KRAS. So that was the low with the lowest IC50 we found against colon HCT116, and the rest molecule. So we picked up this molecule for our further studies. And the last one is the parent molecule of the Boswellia. We did not find much activity against the colorectal HCT116, 1323 micromolar of IC50 value. So further, we did the selectivity index by our semi-synthetic analog of Boswellia. So GD12D was the mutation which was harbored by these HCT116. So there we found that it showed the lowest IC50, and we also had G13D harboring mutation cell line. So SW620, which is harboring G12V mutation and KRAS. So there also it showed good activity. So we also tested the 5-fluorouracil as a standard drug for colorectal cancer, because you know that it is a first line drug for treating tumor. So along with that, we also tested with the at different time points like 24 hours, 48 hours, and 72 hours of the drug treatment by using sulforodamine B cytotoxicity assay. And this heat map, if you see, we did the combination of the our molecule with the 5 standard chemotherapeutic agent 5-fluorouracil, and we could find that the combination index of around 0.35, which is a good synergy between the standard chemotherapeutic agent and our test material. In the heat map we could find, and it has showed a good half-life in the pharmacokinetics. The T half was around 11.96 in the this thing. Further, the in silico study like search of retrieval of interacting genes and protein. If you see the in your right side, there is a crisscross of the proteins where it is basically interaction of MAP kinase pathway proteins. Basically it get amplified by our molecule. So if you see, there is a decrease in the GTP binded KRAS, because GTP binded KRAS is basically get activated in the uh, cancer KRAS mutated cells. So our molecule decrease the overall expression of GTP binded KRAS, and total KRAS has also been reported in the Western blot experiment. Similarly, in the SW620 cell line also, which harboring the G12V mutation, there is decrease in the GTP binded KRAS and total KRAS remain intact. So we further tested the downstream signaling of KRAS mutation, where PI EGFR kinase level has gone down. AKT, we did not see much change, but MAP kinase, we could see the change in the level of phospho-AKT level has gone down. ERK has gone down. So it indicates that it is working through this KRAS mutation, because our molecule is basically helping the standard chemotherapeutic agent, as well as it is helping to downregulate the GTP binded KRAS expression. So we did some molecular modeling studies how it is interacting with the mutated KRAS G12V and G13D, our molecule. So there we could find the stable molecule at the binding pocket site. So we also tested our molecule for the KRAS mutation in the 2D culture, in the spheroid culture of the HCT116G13D model. So similarly, we tested again the how at which phase of the cell cycle basically our molecule is arresting the cell cycle. So we could see that our molecule is basically through G1S phase of the cell cycle, it is arresting the cells. So basically all cell cycle checkpoint inhibitors, we can see the cyclin A2, cyclin E1, CDK, P57 and P21 level has gone up, and all CDK's level have gone down. So this clearly indicate that molecule is basically arresting the cells at the G1 phase of the cell cycle. So what we wanted to do is we wanted to see whether the MGMT, the methylene guanine methyl transferase, which is basically a transcriptional, it is basically doing the transcriptional silencing of these MGMT promoter site. So and restoring the MGMT expression by increasing the mRNA level of the MGMT expression. So basically, it is basically restoring the cells through transcriptionally silencing the MGMT promoter, and basically restoring the MGMT expression in the cells. So we tested the therapeutic efficacy of the PMB and MNU model, like methyl and nitrosourea tumor model in the animal, where polyps in the colorectal cancer can be seen in the picture, where we tested our molecule and we could see the increase in the overall survival time of the animal with respect to the control animals. And there is decrease in the Ki67 proliferation marker in the tumor cells, and decrease in the KRAS level. So that clearly indicate that our drug is basically working through the KRAS mutations, and basically it is helping to pronounce the effect of EGFR kinase pathway by attenuating the KRAS levels and uh, engaging the P50 PI3K signaling pathway, AKT mTOR signaling pathway to induce the cell death in the cancer cells. So in the summary, I would say that the study explored the potential pharmacological property of PMBA, or synthetic molecule of boswellic acid, in a KRAS G13D and G12V mutated profile, and have capability to neutralize the aberrant oncogenic signaling in aggressive KRAS mutated colorectal cancer. Collectively, these results suggest that PMBA alone can be used, or in combination with 5-fluorouracil may be very effective strategy to targeting colorectal adenocarcinomas by with harboring these type of mutations in the KRAS. From the drug discovery point of view, the study unveiled the important molecular association between the transcriptome of individual KRAS mutated colorectal cancer. So these are the some of the leads which we have generated. You can visit the detail about our triple IM drug pipeline, where we have many compounds which have reached to the advanced level of the drug discovery stages. So with this, I would like to close my talk by saying that the road to the clinic goes through the pathologic museum and not through the apothecary shops, by Sir William Whittingall. And this is the picture of Sir Colonel Ramnath Chopra, who established this lab, who was known to be a father of Indian pharmacology. And we all know that in the pharmacy textbook we have studied, in the forensic pharmacy book also, that Chopra committee. So he was the person who pioneered in establishing the regulation of Drug and Cosmetic Act in India during the British time, when the import and export of the drug in was not regulated in India. He was the person who institutionalized all these things. So with this, I want to thank all of you from the Society of Pharmaceutical Sciences and Research, especially Dr. Shashi Alok and his team members, for inviting me for the talk. And I will be happy to answer the queries raised by the listeners. And at the end, at the outset, at the end of the event, I would say that today, you know that today is the World Cup match between India versus Australia, which will be played at Ahmedabad. So I would like to congratulate Indian team, and I would like to give them best wishes from the team, from the behalf of Society of Pharmaceutical Sciences, that we should bring the World Cup to India once again. With this, I thank you all. Thank you.
Thank you so much, sir, for such a wonderful and enriching session, for enlightening us with your knowledge and valuable words. So, before I conclude the session, some questions are being asked by the audience.
Okay.
First question is, how can we detect zero stage cancer? Does it show any symptoms or it is silent?
Usually, symptoms are usually the loss of appetite. It will, like, if you, before going for biomarker testing or advanced testing, if you see some bowel habit changes, or if you see some changes in overall, your appetite, your weight loss, or if you are having regular episode of fever in the evening time, so these are some of the indicators which normally body gives before this thing. So you should not neglect those early signals also uh, before going for advanced testing. So I would say that always go to visit to your physician and tell him about your general health, that these are the issues which I am facing, I am having, without doing anything. If you are losing weight without any crash dieting or exercise, if there is a weight loss, or if you are having episodic fever regular basis, or if there is change in the bowel habit, so these, one should not take them lightly, and they should visit the physician. Because these are the early signs, some of the early signs of cancer. So one should not take it lightly.
So, the second question is, sir, how can we reduce or control the angiogenesis in the cancerous cells?
See, angiogenesis controlling is, there are some drugs which are basically specifically target the angiogenetic factors, like basic fibroblast growth factors, and there are many growth factors which are basically responsible for growth of cancer. So by depriving the cancer, by stopping the matrix metalloproteases inhibitors, these can be stopped, because cancer is a highly metabolic cells, so they need lot of nutrition to proliferate. So they try to get the nutrition from the neighboring tissue, and by releasing these growth factors, they try to branch out from there, this thing, to get nutrition from the neighboring cells. So to stop these cells, some of the angiogenetic inhibitors are there, which can be used if it is required to be given by the physician. They will definitely add into the cocktail so that these angiogenic factors can be stopped to further formation of new blood vessels for the neighboring tissues.
Thank you, sir. The next question is asked by the Prashant Kaushik from audience that he has synthesized a compound and it is showing anti-cancer activity against the lung cancer and breast cancer. It is there in literature. So he's asking where can he perform that activity? Is any collaborative collaboration is available for that?
Yeah, we can test that, but only thing is before that you like to see the novelty and other thing. Like if it has already been reported, it has no meaning. Like if these compound are really novel, so some basic patent search can be done through or some sophisticated tool like Finder,
I
and
find it.
other tools are there where you can just find out whether this is the new compound or first time been reporting your.
So,
So
sir, if it is, if it is found, it is clearing that novelty test. So, can we? What is the process in the charges like that for the testing? What is the process through which the person can enroll?
see, there are two ways of doing. If we have a collaborative work, we can do it on our own basis, but on sharing the intellectual property. But if it is through payment basis, then you can pay. You not to share the intellectual property with us. So both way it is possible. Like if you want to share the IP with us, then both of us can do the work together, and we need not to charge you for that. But if you want to do it for you don't want to share your IP, then we can do on payment basis. And payment, this thing is available on our website, that what are the charges. You can just visit the rate list where you can find out.
Okay, sir. The next question is, how does precision medicine contribute to tailoring cancer treatments to individual patients? So, are there some specific examples of successful applications of precision medicine in cancer drug discovery?
Because you know that PDL1 receptor, when it was discovered in 2016, and they got Nobel Prize for PDL1. From there, the immunotherapeutics started. So PDL1 is one of the receptor where the immunotherapeutics are used, pramzolimab and other drugs, which are being Yorley. These are the drugs which are commonly being used in US and other developed countries. Though right now they are very expensive, so it is very it is not easy for a Indian patient or any other poor country patient to afford these kind of drugs, because either they should be subsidized by government or by some other agency. They can be available to the common man, but they are being used and they are very successful because you don't face lot of side effects, because they are very precisely targeting the cancer, because they work through a mechanism. If that receptor is present on the cancer tissue, definitely it is going to work. But if it is not, like as I have told you in my lecture, the Kras mutation, and they will check for EGFR therapy before giving the erlotinib. They will check the Kras status. So if Kras is mutated, they will not go for EGFR therapy, because the downstream signal will not pass through, because in between there is Kras sitting. So if EGFR upstream is EGFR is there, so downstream signal will stop at Kras. So that will not, it will give a very poor therapeutic response. So testing these biomarkers are very important for precision medicine. And now the era has come, there is a paradigm shift in the treatment plan, that it is becoming more individualized treatment for cancer patient, to go for genome sequencing first and identify your bio mutations which are present in your cancer, and then you can take advice from doctor. They will decide the which drug will be most suitable for this type of cancer.
Okay, sir. Thank you. The next question is, how can one use the SEPbox technology in your esteemed organization? And what will be the cost in case of the academician? Is it different for the industrialist and academicians, sir?
Actually, Sabbox, I don't know what is the current status, but Sabbox is basically for samples where you have all parameters are set, like you have a HPLC method developed, all other parameters, so that on that basis you can easily use that. So one has to get into those detail, because any fraction or any isolating the pure crude extract will not be helpful for that. First he need to have a standardized extract, and some identified markers should be there, so in that basis he can use that.
Okay, so the next question is, what are the major challenges that are faced by the researchers in the current landscape of cancer drug discovery?
Major limitations.
Yes, sir, challenges or limitations we can see.
See, limitation is the resources in the like some of the labs, like initially this cell culture this thing facilities are expensive in nature. If you store the cells in your lab, you need lot of plastic ware, they are expensive. The media, growth media, the FBS, fetal bovine serum, is costing a high value, like 1 lakh rupees 500 ml bottle. So if you have to buy the US origin FBS, so still the costing is one of the reason and one of the limitation, I would say, for discovering the drug, because you need to do all cell line based testing first, then you need to set up the in vivo tumor models, and tumor models are also not easy, like because you need a nude mice, skid mice, and they are very immunocompromised mice, so you need to cage them in a individualized ventilated cages, and these cages are expensive. Then you need gamma radiated feed for them, you need autoclaved feed, autoclaved bedding, gamma radiated feed, so all these are expensive in nature. So overall cost will go up for a common laboratory to bear the cost. So I would say this is one of the limitation. And of course the chemicals which are used in the biological science, the antibodies which are available commercially, they are also expensive. And then further testing, like once you have established all the preclinical data, still for getting the approval for clinical trial and other thing, that is also not very easy, this thing. So I would say the rather than doing this repurposing of drug is better approach for discovering new molecules.
Okay, sir. Sir, the next question is, what is the correct method to calculate the IC50 values in case of anti-cancer drugs? And as we change the method, what method should be used?
IC50 is usually like there are different methods commonly used, like sulforodamine BXA, XTTSA, MTTSA. So all are having different kind of principle. Like MTT is basically measuring more viability, whereas if you see the sulforodamine BSA is basically measuring the cytotoxicity. In a way, both are same only, but they basically in MTT you see the pharma zone crystal formation, and if the cells are more metabolically active, the color will be developed. Basically, both are colorimetric assays, but in the sulforodamine BSA, which we do in our lab, is basically uh, bind to the dyed cancer cells. So you dissolve in the tris buffer that amount taken by the cells, live cells. So that that way both are colorimetric in nature, but you need to have a non-linear this thing relationship, because you will not have a linear kind of regression over there. So you need to identify the linear part in the regression analysis. From that basis, if you have a concentration versus your cell growth inhibition plot, where you can have a initially you have a linear kind of uh,
pattern, then you have a hyperbolic kind of pattern. So you will need to pick up the R value 0.9 or 0.8. From there, you can calculate the IC50, that C versus means your concentration versus your growth inhibition. So in the non-linear regression analysis, you can calculate the IC50s.
Thank you, sir. So the next question is, what stages does a potential cancer drug go through in the development process from discovery to clinical trials?
I could not hear you properly. Can you just repeat it?
Sir, what stages does a potential cancer drug go through in the development process from discovery to clinical trials?
See, there are many phases, like some drug may get quick authorization, because if there is a orphan status, or if there is no drug available and for that particular type of cancer, they may get far accelerated approval. Like Bortezomib got 7 year time to get approved for multiple myeloma cancer. So some drug take 20 to 15 years. 15 years is a usual time, that for drug discovery, right from the preclinical to clinical approval, if everything goes well. But in case of if you talk about fast track approval, sometime if FDA feels, regulators feels that there is need to have a drug in this class, because there is no treatment available, there is a urgent medical need is there. So in that situation, they may approve within a short period of time.
Thank you, sir, for your valuable time that you have spared and so much knowledge you have shared with us. It was really a wonderful session. We have gained a lot through this session.
Thank you, Dr. Payal. Thank you,
So,
Mrs. Dr. Monica Sabarwal. Thank you, Dr. Shashi Alok, for inviting me. Thank you very much.
To conclude the webinar, we would like to express our heartfelt gratitude to to our esteemed speaker, participants, and guests for joining us today. Your active engagement and insightful contributions have made the event truly enriching and valuable. The discussions led by our esteemed speaker, Professor Dr. Shashank K. Singh, have been both enlightening and thought provoking. Throughout the discussion, Professor Dr. Shashank K. Singh has discussed on various subtopics such as target-driven drug discovery from natural products, conservation and production of bioactive plants using in vitro techniques, standard grade treatment and biomarkers for cancer, currently approved latest cancer treatments options, personalized care in the cancer, and the role of radiotherapeutics in cancer. The expertise and knowledge shared by our distinguished speakers, Professor Dr. Shashank K. Singh, Dr. Shashi Alok, Mrs. Monica Sabharwal, and entire SPSR committee have been instrumental in shedding light on this cutting-edge approach and making this event a successful. I encourage each one of you to carry forward the knowledge gained today into your respective roles and fields. Whether you are a researcher, clinician, or industry professional, your contributions play a vital role in the ongoing fight against cancer. Together, we can contribute and continue in advance to the frontiers of science and bring about transformative change in the landscape of cancer drug discovery. Thank you to our esteemed speaker, organizing committee, and most importantly, all of you for your engagement and commitment to advancing our understanding of cancer and its treatment. Let's remain inspired by today's insights as we collectively strive for a future where targeted and personalized treatments bring hope and healing to those affected by this challenging disease. Have a great day. Thank you, sir.
Thank you. Can I leave now?
Yes, sir.
Thank you. Thank you very much.